소개 감사드리고요. 예, 저는 김종민이고요. 저는 포항공대 생명과학과에서 2018년부터 일을 하고 있습니다. 예, 그래서 제가 오늘 말씀드릴 내용은 제가 이제 주로 포스닥 때 그 위스 연구소 하버드대학의 위스 연구소에서 했던 일인데요. 이 부분에 대해서 말씀드리도록 하겠습니다. 예, 그래서 일단은 영어로 준비했기 때문에 이 다음 장부터는 영어로 진행하도록 하겠습니다. Okay, so let's talk about synthetic biology. So what is synthetic biology? Synthetic biology is about building biological systems. So this is one of my favorite image. So Lego man. So synthetic biology is about like building biological systems. So through the process of building, we can explore beyond what's available in the nature, and then we can also learn something about the biological system and the mechanisms of it. Okay, so we can think of synthetic biology as a redesign of existing natural biological systems for useful purposes, and also for the design and construction of new biological parts, pathways, and devices and systems. So we think that synthetic biology will become very important for the coming bioeconomy. So bioeconomy is about economic activities fueled by the research innovation in the biological sciences. So synthetic biology will become play an important role in the bioeconomy. So including the food areas and also biomass and also climate change. And another thing that people are really interested in is about the microbiome. So microbiome is a very, very hot topic because there's more and more evidence that the microbiome is connected to the human health. So one of the application area of the synthetic biology can be engineering the microbiome of the humans. So to do that, people are thinking about building a smart probiotic system. So as illustrated here, so you can think of it as like a subnormal bacterium that's normally living in our system. But then you can think of it as expanding those functionalities by having those sensors such as drug and quorum sensing molecules, and also sensing the disease markers, and then integrate those signals and then do appropriate action like releasing drug, and for example, or stopping the replication or controlling the population of it. So we'll mainly talk about synthetic gene circuits here. So this is a somewhat fancy diagram was used in the MIT News back in 2013. So here you have a bacterium, but it's having a CPU on board. So it's like so what we really want is having these kind of complex functionalities utilizing those computer systems like in the bacteria. So first, let's talk about logic gate. So AND logic gate is illustrated here. So it has two inputs and one output. So AND gate is true when both of the inputs are true. So when A and B are true, the output is true. And we can do analogous thing in the synthetic biology system. So in this case, you have two proteins, SIG-A and INVEV, and those two proteins form a complex and then drive the expression of the downstream gene. So in this case, you can see that you have to have both of proteins to be high to be able to have a high GFP output. So it's an analog of the AND logic in the synthetic biology systems. So you can build a pretty complex system using these kind of protein-based logics, but there are some limitations. So what are the limitations? One of the limitations is that usually you're mining these parts from different organisms such that those different proteins are not necessarily designed to work together. So some of them can recognize similar DNA sequences and drive the expression of the gene. So it can be crosstalk among parts. That can be a big problem. So you have to check for the crosstalk. Another problem is that if you want to build a complex system, usually you have to assign different proteins for different gates that you have designed. So you have to assign distinct proteins for each gate, so which means you can run out of the parts pretty quickly if you want to build a complex system. So the goal we wanted to test was, can we design a novel regulatory parts from scratch? So can we design novel parts from scratch? So this is an example of the novel regulatory parts using RNA. So back in 2004, Jim Collins group used this design. So you have a ribosome binding site that's hidden in the stem structure, and then you have a loop, hairpin loop, and then you have the start codon and downstream gene. So in this state, because ribosome binding site is hidden in the stem, you cannot express your downstream gene. But if you have a trigger sequence that's illustrated here, you can start binding from the hairpin loop, and then you can open up the rest of the hairpin stem. So you can now express your downstream gene, so which is fine. And they were able to demonstrate a couple of examples. But then the limitation is that you have a strong sequence constraint. So because of the YUNR motif in the loop, you have to have the same appropriate sequence, complementary sequence, and then you have to have RBS-like sequence to be able to open this loop. So which means that you cannot build many devices using this approach because of the strong sequence constraints. So back in 2014, so I was working as in the Peng-In group as a postdoctoral scholar, and then Alex Green, then at the Peng-In group, decided to change this design. So now you can see that it looks different. This is also riboregulator, but then now the ribosome binding site is in the loop, hairpin loop, and the start codon is in the bulge of the stem. So, and then these are binding domains. So you can see that the trigger RNA has a binding sequence here, but then there's no color, which means that there's no sequence constraint. So now you can design many devices without worrying about the sequence constraints. So let's look at the mechanism of Toehold switch through a short video. So let me play it here.
The Toehold switch is an RNA that encodes the gene for a protein of interest. However, translation of the gene is initially turned off. A hairpin structure in the Toehold switch hides the ribosome binding site and the starting sequence for the gene, and thus represses production of the protein. The Toehold switch is designed to detect a target RNA. The Toehold switch contains a sequence that is perfectly complementary to the target RNA in the early half of the hairpin. When the target RNA is present, the Toehold switch recognizes and binds to it. This binding opens the hairpin. As a result, the initially hidden ribosome binding site and the starting sequence are exposed. The reporter protein can now be expressed.
Okay, so yeah, I hope you can get a better idea about the mechanism of Toehold switch. So essentially, we are designing a secondary structure where the RNA can bind, and then when the RNA binds, it can allow the access of the ribosome, and then you can actually translate the downstream gene. So what do we do? So to actually implement this in E. coli, you can actually design RNA sequences, but it can be designed de novo, which means that you can design from scratch. So there's no, this is not a natural system. We are designing an artificial riboswitch, switch, so you can use any sequences that you want. So you can design from scratch, for example, using a computer. And then you can put a trigger in one of the plasmid, and you can put a switch and a GFP in another plasmid, and you can transform both of them in E. coli and see whether it can work. So let's look at like a flow cytometry data. So this is autofluorescence in the GFP channel. So there's some basal fluorescence in the E. coli. And then if you have a switch that's connected to the GFP, the fluorescence level is about the same as autofluorescence, which means there is almost no background expression, background leaky expression. But once you have a switch and a trigger at the same time, you can actually see a great increase in the GFP fluorescence, and which is very similar to the positive control, which is just GFP. So which means that we are really be using this synthetic riboregulator, we could exploit the full dynamic range encodable in the E. coli. And then there's a clear difference between on and off states. So because there is no sequence control, you can actually build many devices and test them. So for example, Alex Green was able to build 168 different Toehold switches and then test their performance. And some of them are very good, as shown here, and some of them are not so good. But in the process of this testing and building these systems, you can actually learn something about what is the important motif, sequence motif you have to use to build a good Toehold switch. And then it also has a good orthogonality. So previously we mentioned that in the protein-based regulator, having a good orthogonality, which is having low crosstalk, is important. And then here you can actually see that there's almost no crosstalk in the switch and trigger indexes. So these are cognate triggers and switch pairs in the diagonal. These are non-cognate pairs, and there's very little crosstalk. And then it was exceeding the existing the RNA-based regulator much more. So these were the existing RNA-based regulators at the time, and these show the dynamic range and library size of the Toehold switches. And then you can also use it for sensing endogenous RNA. For example, the RIB is a ion-responsive small RNA that's important for the bacteria, and then you can design a Toehold switch around it so that you can have a RIB sensor, and then you can actually see the expression of the RIB to be monitored through the expression of GFP. So to summarize, so Toehold Switch is essentially detecting RNA sequence A and then make a protein sequence B. And then there's a good dynamic range and also has good orthogonality. And then you can just program it using computers because it's a not natural system, it's a synthetic system. And then you can actually use arbitrary sequences if you want. And there can be some applications. For example, you can use the Toehold Switch to sense messenger RNA or some other endogenous RNAs. And you can also use it for computation links, which will be the focus of the rest of the talk today. So we'll talk briefly about the ribo computing device, which is essentially a generalization of the Toehold switch. So we published this result back in 2017. So here you are seeing a Toehold switch-like motif, but there are several of them in a single RNA transcript. So there are sensor module, and then also RNA network can be detected as an input, and then you can also have a same output gene as your output. So how do we design this? So similar process as a Toehold switch. So you have a secondary structure that has RBS and start codon, and then you can assign sequences using computers, and then you can actually test them by connecting to a fluorescent protein, for example, GFP. You can test for performance, and then let's say you have a number of these validated switches, Toehold switches, then you can list the validated parts, and then you have a number of them, and then now you can consolidate those parts into a single long RNA. So now it is what we call a ribo computing device. So why do we want to do this? So for example, let's think about a two-input OR gate. So this is what you want to build, what you have. And then let's say you want to build a 6-input OR gate. Then you can use multiple two-input gates to build this system, but then you have to use up a lot of those parts. But what if you can just build a big 6-input OR gate to begin with? Then you can potentially scale up the system complexity much more by using these kind of bigger gates. So that's the motivation for having this multiple sensor domains in a single RNA transcript. So this is what we call a rate gate RNA in the ribo computing devices. So you have multiple sensor domains, Toehold switches. And then now each of them has a ribosome binding site and a start codon. So each of them have individual ribosome binding site and start codon. And then you have the cognate input triggers, let's say A, B, C, D, E here. And then they can be each recognized by these color-coded regions. But then what you notice is that let's say A binds here and then open up this hairpin, then the ribosome binding site is exposed. So ribosome can come in and then start the translation. And then because of the ribosome have a great processivity once you start the translation, then you can actually go through a number of small secondary structures, even though you cannot initiate when at this structure. So essentially what you're doing is that you're performing an OR logic because any of the input is sufficient to open up the ribosome binding site and then allow the translation of the downstream gene. So let's see how it actually works. So let's say you have a 2-input OR logic. So you have the device to detect input sequence A and B. So A and B. So let's say you have A, then A binds and then you start the translation. And then since you have already started, you can actually go through this structure and then translate your downstream gene. But let's say you have input B, then you open up the second hairpin, then definitely you will express the downstream gene. So either of the input will be sufficient to translate the downstream gene. So this is actual experimental result. So you have a non-cognate RNA, then you have background level of expression, so which is autofluorescence. And once you have A, then you have a pretty good expression. Once you have B, you have also a pretty good expression. And you have both, then you will have even more expression. So if you quantify it, it looks like this. So you have no input, or one input A, or one input B, or you have both. Then you can actually get a pretty good expression, which is much like an OR gate that we talked about. Sorry for the notice. I should have turned it off. Yeah, sorry. So and then we can actually expand this into a 6-input OR gate. So now you have 6 messages that you want to detect. And then you can actually test them in the flow cytometry. So if you have a non-cognate RNA, then definitely there's no significant expression. But if you have any of the input A, B, C, D, E, F, A, B, C, D, E, F, then you can actually get a pretty good expression. So this is the result of the quantification. And then you can see that you have a pretty good expression for all the inputs, and then almost no expression for all the non-cognate inputs. Because it's hard to see, you can actually put in the log scale to make it clear that there's almost no expression for the downstream, for the any non-cognate inputs. But then, as you probably have noticed that there's actually some limitations. So for the input sequence D, E, and F, you have a pretty good expression, but not as good for the A, B, and C. So which seem to indicate that you can actually go through all the downstream hairpins, but sometimes you can fall off. So you seem to say that, okay, if you want to trigger far left, like using A, then you can go on, but sometimes you actually fall off in the middle. So there is some limitation for the activation of the leftmost side, but we are still working on it to improve this. But at least we were able to show that we can have a functional 6-input OR gate that has about 100-fold on-off ratio. And then, so we talked about OR gate, but what about an AND gate? So we can think of it as this. So now you can think of this input RNA as conceptually as two different domains. So the 5' part we can think of it as C1 and 3' part as C2. So if you have a full length input, then definitely you will be able to turn on the system. But let's say you have only a 5' part. Then 5' part wants to invade into this stem, but there's no way to invade it easily because it's already closed as a stem. And if you have a 3' part, 3' part can bind to the single-strand region as a toehold, but there's no invasion part, so there's no change in the structure. So which means that either C1 or C2 is not enough to activate the system, and you have to have both of these domains. But since we are talking about RNA, you can easily design a structure using Watson-Crick base pairing. So let's say you have a 5' domain here and a 3' domain here. And then you can actually design complementary domains. This is color coded. So let's say you have these complementary domains. Then it will actually be able to self-assemble into a structure like this, which presents the 5' part and 3' part in a close proximity. I mean, even though it's not a single strand, because these two domains are in proximity, it will be able to hopefully activate the system. So now we have all 4 of these strands, and then they come together and presented as a single complex. And then now we need all of them, so it's more like an AND logic. So let's think about a 2-input AND gate. So you have a 5' part here, and then there's a connecting domain termed U star, and there's a 3' domain, and then also connecting domain U. Now these U and U star form a bridge like this. So now you have a complex that presents the 5' part and the 3' part in a close proximity. And these two together can hopefully turn on the switch. And then this is experimental data. So if you have no correct input, non-cognate inputs, then you definitely have no expression. You have either A1 or A2, then there's not enough to express the system. But if you have both, then you can actually have a pretty good expression. So now it works mostly like an AND gate that we designed. So this is a 2-input AND gate, and this is the best one that we could find. And then we can actually scale up into a more complex system, such as a 4-input AND gate. Now you have a 5' domain here, 3' domain here, and then 4 of them come together into a complex. So what you notice that is it is functional, but then the performance is much worse compared to the previous 2-input gate we talked about. So there are some limitations. So maybe there is some misassembly because we try to make a rather complex system in living cells, and also there might be some mismatch in terms of the stoichiometry or like some misfolding potentials, or also entropic penalty because we are putting a lot of the small strands into a small space. So there is definitely more work to be done to improve the system, but we were able to at least demonstrate that 4-input AND gate was at least functional with about 9-fold on-off ratio in the E. coli. Okay, so we talked about OR gate and AND gate, but what about NOT gate? So NOT gate, the simplest thing you can think about as NOT gate is using antisense system. So you have a trigger RNA, but now we have elongated a little bit on the 3' and 5' end using U and V here. And then now you have an antisense, which we call as like input B. And then the antisense has a longer domain, binding domain, so you can use these U and V as a toehold domains to actually strip off the input if input was already bound to the switch. So because of the thermodynamic advantage, it will be able to strip off this input. So now you should have the input, but not the antisense to be able to activate the switch. And you can use this idea to make a more complex system. Let's say something like this. So you have RNA sequence A1 and A2, but not the antisense for A1. Or you can have a B1 and B2 and not the antisense of B2, for example, etc. So what do we mean? So this is what we mean. So we have AND gate in the trigger we talked about, and then we have antisense also in the trigger side that we talked about, and then the OR gate was built into this gate RNA design where you have multiple Toehold switches. So now you can actually funnel in all this and then not close into OR gate. And let's see how it actually works. So this is a flow cytometry data, so where you show that these are all the non-cognate input cases. I mean, so or the false cases, and then these are the true cases. So let's evaluate this. So let's say A1 and A2 and not antisense A1. So 110 has a good signal, but not all the other cases. And B1, B2 and not B2 star. Similarly here. And then C1, C2 or D1, D2 or E1, A2. Yeah, so this is a summary of the data. So I mean, because since we talked about 12, ideally we should have evaluated all the 2 to the 12th cases, but there's too many, so we just evaluate for each individual gate. So we all tested all connections for the combination for the A1, A2, and A1 star, B1, B2, and B2 star, and C1, C2, D1, D2, and E1, A2. And these are all the correct input combinations. So we were able to demonstrate that a 12 input gate can be functional in E. coli with about 20-fold on-off ratio. And also another question is, can we actually operate more than one of these ribocomputing devices in a single E. coli? So we decided to test this case where you have a GFP output after a single 6 OR input gate, and then also you have M cherry output after a 5 input OR gate. And you can actually test the performance in the same cell. So let's say you have the M cherry input, and you actually have a pretty good expression of M cherry. You have a GFP input, you have a pretty good expression of GFP. But now, as we mentioned, that this is not perfect. So because if you want to try to activate from the far left side, the output level is not as good, but it was at least functional. So you can test the individual M cherry input or GFP input, or you can have M cherry input and then another non-cognate input, GFP input and another non-cognate input at the same time, or you can have both M cherry and GFP inputs. And then you can actually see that because the E. coli is ordered to express very high levels of both first proteins, then there's some metabolic burden, and they could not achieve the same level of expression, but it was at least able to show that you can actually control both of these outputs successfully. Okay, so yeah, we talked about the ribocomputing device, and then we also have going to talk about the main topic of the today, so which will be about designing the repressor system using these Toehold switches. So fortunately, it was selected as a cover image thanks to the nice image provided by Alex. So this is about using same idea, so using the controlling the accessibility of the ribosome to the RNA, and then you can actually compile the complex logic, implement the complex logic in the E. coli. So this is one of the design Toehold repressors. So this looks somewhat different. So you can see that now you have a bigger hairpin on the 5' side, and then the ribosome binds side and start codon is open. So this is the active state. So ribosome can come in and then translate fine. So normally you are in the on state. But then let's say you send the trigger, there's a little bit long ABC domain, then you can actually bind here and then open up the rest of the hairpin. And then now because of this red region, the B prime, and then this red region, you actually form a structure that's analogous to the Toehold switch that we just talked about. So now once you have the trigger RNA, then you actually form a Toehold switch-like structure, which means that your translation will be off. Also, there's another design that we designed. So this is a switch RNA that we talked about, but now the bottom stem is very weak, so it's almost open. So bottom stem is very weak. So normally ribosome can come in and translate fine. So this is the on state. But let's say you send a trigger RNA, which has this conformation, then you can actually come in and zip up these A star and B star domain. So now you form a stable three-way junction. So now you cannot actually translate your downstream gene. So once you have the trigger, you actually form a three-way junction and then you stop the translation. So this is another design. And then you can actually, because as you have seen, there's no sequence constraint here, similar to Toehold Switch, so you can actually design a big library and then test their performance. For example, we could construct a 96 member library and then test the performance. And then some of them have pretty good performance, like more than 100-fold, or some of them even have about 400-fold repression compared to the not having the trigger. And then you can also use this as a sensor, similar to the previous tools we talked about. So now let's say you want to detect a messenger RNA, then you can design a toll repressor that can sense those regions, or you can also have a three-way junction repressor that can sense those regions, and then they can actually stop. These RNA can be used as inputs to stop the translation of downstream gene. And then you can use this to you can use some of the antibody resistance genes as an input, like a kanamycin resistant gene or beta-lactamase gene as a messenger RNA as an input, and then you can actually see that it can actually perform the sensory role in the E. coli. Or also similar to the rival computing device we talked about, we can actually connect more than one of these repressors into a single gate RNA, and then you can actually use it as a multi-input devices. So let's say you have these two 3A junction repressors connected as a single gate RNA, and then you have input A. It will bind and lock the first one, but then since the second one is still open, ribosome can come in and actually translate bind. So you have to also have both second input to lock the second structure. Then you cannot actually now express the gene. So it's analogous to a NAND gate, which means that you have to have both inputs to not express your downstream gene. So this is the result. So you have no input, you have one input, then you can actually have a pretty good expression. But if you have both inputs, then your expression is very low. This is like a two-input NAND gate. And we can also try to scale them up into a more complex system, like four-input NAND gates. This is a similar idea. So you have more. Now you have 4 different three-way junction repressors that are connected to a single RNA. And then now you have to have all of those inputs to actually stop the translation. And that is the result. So as you can see, the performance of this four-input gate is not as good as two-input NAND gate, but still you were able to demonstrate it was functional as a four-input NAND gate. And then also, what was important to us is that you can actually swap those order of these gates pretty easily. Let's say you have a 1390, this is just a numbering of the index of those from the library. Let's say 13 and 19 or 13 and 24, 11 and 13, 13 and 10. I mean, there are a number of combinations that you can use as a two-input NAND gate, and then those were mostly functional. I mean, some of them are better than others, but what's important is it was mostly functional. So we could easily swap those elements. And we actually have also different combinations, and this is the one that we show as a main figure. And also similarly for the 3-input and 4-input NAND gate, you can actually have a different ordering, and then without worrying too much about the performance. So sometimes it's not as good as shown here, but in most cases it was pretty good even when we swap the orders. And then on 4-input and NAND gates. So to summarize, so we talked about this rival computing devices. So this is an illustration of the rival computing device where you have multiple sensor elements in a single RNA strand. And then this is, to our knowledge, the most complex logic demonstrated in bacteria. And then because we are using these short hairpin structure as a sensor, it has low genetic footprint. So each of these rival computing sensor has about 100 base. So each of these hairpin has about 100 base long. So compared to a protein regulator, which is typically a few kilobases, so we can actually have a very small genetic footprint in terms of designing a complex system. So because of that, we believe that it will be more scalable in terms of building complex systems in the synthetic biology applications. And since we are relying just on the Watson-Crick base pairing of the nucleic acids, we can be a general principle. So, I mean, Watson-Crick base pairing will work not just in E. coli, but in other bacteria, and also in even outside the bacteria. So it can work in vitro. So people have been able to use this Toehold switches and outside the bacteria, like on a paper as a sensor modules. So, and then there can be some future directions. So using this rival computing device, we can actually try to build a more complex circuit, as we talked about, and then also we can probably use it for smart products, because hopefully we want that it will be functional in other bacterial systems without a problem. And then maybe we can use a similar principle for the eukaryotic system. So it will be nice, but definitely there will be a lot of challenge, because eukaryotic system is very different in terms of translation regulation compared to prokaryotes. But we hope that some of these principles maybe we can port them into eukaryotic systems. And then, as we mentioned, it can be used as a biosensor diagnostics, for example, on a paper. And then we we couldn't have done this without all the collaborators here. So definitely, I'd like to thank my co-doctor mentor Peng In, and then Jim Collins and Pam Silva as the collaborators, and then a number of people at the Peng In's group, and Alex Green, who is now at the Arizona State University with his own group, and then also people at the TUM and so TUE and also Northwestern University. So thank you. 감사합니다.